Hello, all you cool cats and kittens, and welcome to Unit 4, Cell Communication and Cell Division. So here we're going to talk a little bit about how cells talk to each other, both at the cellular level and a little bit at the organismal level. And then we're going to talk a little bit about what controls cell division. So let's start off with cell communication. All right, so first off is let's remember that cells can communicate with each other. And I want to go a little bit simplest form. And I didn't get too much of a chance to talk about this, but I want to talk about our bacteria or our unicellular organisms. So how many of you guys have gone to the dentist office? I'm sure most of you guys have. And you have gotten your teeth cleaned out really well by that plaque that builds up on there. And we know we're supposed to go to the dentist to make sure we clean up that plaque at least twice a year. What some of you guys may not realize is that plaque is a form of communication that's going on between the bacteria in your mouth. And really what's happening is you have what's called basically a biofilm. On your teeth you have what's called a biofilm. So what is a biofilm? Basically it is your bacteria shown here, in blue. You have some maybe other cells going on. You get some DNA, you've got some enzymes, you've got some other enzymes all here. You just got this milieu of stuff building up on your teeth. Like I said, there's definitely bacteria in it. And what happens is these bacteria are actually going to communicate with each other through this film that is on there. And what that film is going to do is going to help it to talk to each other. This is how they can find suitable, change metabolic rates, or talk to each other about how many organisms they are, because at one point they are going to get rid of some of their cells and pass it on elsewhere if there's too many of them. These are things that are going to change and affect how the body works in some ways. So we call that quorum sensing when they are talking to each other through that film area on self. But that's also why it's so important that you do clean your teeth. We do know that if you don't clean that plaque and stuff like that, some of these cells can come out and actually affect other parts of your body, including your heart. So these are important aspects in terms of why if you have a heart murmur, if you go to the dentist, you're supposed to take antibiotics beforehand because they want to kill off that bacteria. So if they do clean things out, these bacteria don't go and affect your heart even more. Things like that. People who have catheters have to worry about a biofilm forming on the catheters all the time as they take it out and put in a new one in there and stuff like that. So again, it's a different way of us looking at bacteria and unicellular organisms because they do communicate with each other and they do form these biofilms that can affect our health in different ways. Now, we can also have cell communication, but this can be at the organismal level. So one way that cells can talk to each other that affects the whole organism is something called ataxis. Basically, that is any movement of an organism in response to a stimulus. So what do I mean by that? If there is a stimulus, so gravity can be a stimulus, then the organism is going to respond to that gravity. And that is something we see here. Fruit flies, and I know you guys love my fruit flies, they have a geotaxis, which basically means when they sense gravity pushing down on them, they're going to go in the opposite direction of gravity. So let's say gravity is going down, and you should see that they would go opposite of that, which is up, every time they have the effect of gravity. Now you should be able to notice that shown here, as they're younger, they sense it better. As they get older, they have a hard time sensing it. 4 weeks old. Yes, fruit flies can last 4 weeks. Can you believe that? Because these fruit flies are going opposite the direction of the stimulus, this is considered a negative geotaxis. So it's going opposite the effect of the stimulus. If it's positive, that means it's going to go toward the stimulus. So you can have an organism that has maybe a negative geotaxis. That means it's going to go up. So geo means it's in relationship to gravity. That same organism can have a positive chemotaxis. Chemotaxis means it has to do with smells or chemicals. Remember, smell is a chemical, taste is a chemical overall. So it can go toward a specific smell. That would be a positive chemotaxic. At the same time, you can have a negative chemotaxis where a smell can be repulsive. Phototaxis means phototaxis means it's light. So again, fruit flies actually has a negative geotaxis, but a positive phototaxis means they also tend to go toward the light. And then lastly, thermotaxis is something like temperature. There are actually a lot of other taxes. These are just some examples that you can hopefully figure out based on the name. You don't have to know all of them. I was just kind of giving you examples, so if you saw it, maybe in the word, you can figure out what those words are. I cannot expect them to use a taxis that has a word that's harder to figure out without them explaining what that type of taxis is. But if you got any of these and they don't explain it, I'm hoping you'd be able to figure out using your root words what each one of those are. Now, we saw with the unicellular organism that they were able to talk to each other using the biofilm. Multicellular organisms, the cells, one of the difficulties when you have something that's multicellular is you have to coordinate all the communication that's going on through all the cells within that organism. You don't want your brain doing something and your legs doing something different, basically, or your heart doing something different, or your red blood cells. So what has to happen is you need to have a great communication system for all those cells in a multicellular organism to talk to each other. Some of this can be short range, one cell talking to the next cell right next to each other. Something like we see with a neuron even. Or some of it can be long range where we have your cell releases a whole bunch of chemicals and the other cell that picks it up is really, really far away, so we have to kind of go into the bloodstream to get it eventually to where it needs to go. The biggest thing that you need for this communication, that you need no matter whether it's long range or short range, is you need a receptor, and that's basically going to be a protein that will pick up the signal. It needs to be specific to that signal. And the other thing you need is the ligand, which is that primary messenger or that first signal. So this cell is sending out a message, shown there, that's your ligand. This cell is sending out its message, and again, that is the ligand. When the ligand binds, we can get more messages communicating inside, but really, for any message to occur, we need that primary messenger, which is the ligand, that has to bind to the receptor. Now, one thing we have also discussed is the fact that we have two types of ligand. The two types of ligand can be polar and nonpolar. The polar ligand is going to bind to a receptor on the plasma membrane. And that's really what we're seeing kind of over here. We have a receptor on the plasma membrane. That's what we saw in the last picture. So we're going to have our ligand is going to bind to a receptor. And that's going to send the signal in a signal transduction pathway that occurs into the cell. I'll get into signal transduction in a second. When we talk about, though, nonpolar, the nonpolar, the receptor will be inside the cell. Because that means the ligand can get through the plasma membrane and is going to bind to the receptor inside the cell. So that's one thing to kind of keep in mind when we talk about cell signaling. But basically, overall, what happens is once that primary ligand binds to a receptor, the next thing is we're going to activate that signal transduction pathway. And one of the reasons why we do this is going to help bring a cellular response, but it helps to do a couple things. it will help to amplify the response. So that's one of the reasons why we do this. And it also is going to help to really make sure that the signal is what we want it to be done. So we're not making a mistake because there's lots of players involved to ensure that we really want that response. Okay, so basically what ends up happening is when you have a ligand that binds to that first receptor, it's going to change shape so that the second messengers can respond, and they're going to also activate more receptors, and eventually it's going to cause a cellular response. And some of that cellular response might be increased gene production or protein production. Maybe we need to decrease a protein. But basically all of these things are going to go to the nucleus and it's going to help change what enzymes, what receptors, what molecules we have made in the cell. And there are three main types of signal transduction pathways. Again, these are usually all for polar ligands. But basically what we're going to have for our signal transductions, we're going to have the G protein coupled linked receptors shown right here. We can have a ligand gated ion channel, and then also we can have our receptor kinases shown here. What they're basically going to do is the G protein coupled receptor is going to have a bind a ligand to the receptor. It's going to then activate the G protein, which is internal in the cell. And then what that does is the G protein is going to activate adenylate cyclase, turns on another second messenger called cyclic AMP, also known as CAMP, which then opens up a protein kinase pathway. Remember, kinases adds phosphates. Okay, and that by adding all those phosphates, that's eventually going to activate a molecule that's going to turn on, open up the DNA so we can make protein to change what's going on in the cell, basically. And I'm not very specific here because there's so many different pathways that these G proteins can work. And when I talk about the protein kinase, it changes the DNA, it makes targeted genes. This protein can activate stuff or it can actually turn off stuff. So that's why I'm not being really specific here. Ligand-gated ion channels, basically what happens is a ligand will bind to it, allows it to open up so that molecules can come in. In this case, we're bringing in calcium. Calcium is a very important second messenger in our bodies. And what ends up happening is that's going to activate different pathways. We have two different parts here where we're seeing the pathways here, like this is a calmodulin kinase. And that's going to, again, either turn on targeted genes. Those genes can either activate or turn off pathways in the cell. And the last one are the receptor tyrosine kinases. These are the ones that I always joke around with. That reminds me of the Wonder Twins because they start off separate, but once they bind the ligand, they come together, they phosphorylate each other. So it's not shown here, but they end up activating each other. And that activation eventually causes, again, a second messenger to get activated and again change the DNA so that we're making a protein to either stop or start another function in the cell. So I talked about how these proteins that are being made through these second messengers can either stop or start a function. What are some of the things that we can control with messengers? Well, what we're going to do is what we basically do is homeostatic mechanisms. We're going to maintain the homeostasis in the cells. There's two major loops that we deal with in terms of homeostasis. There's the negative feedback loop, which is the most common form. There's very few of the other one, which is the positive feedback loop. Now, hopefully you remembered when I talked about the negative geotaxis or negative taxis, it's opposite of what the stimulus is. And that's the same thing with a negative feedback loop. What happens is in a negative feedback loop in your body, you get a stimulus, it gets picked up by your sensors, then ends up getting sent to a control center, that produces something to affect it, and that effector is going to turn off that stimulus. Okay? That's a negative feedback loop. So one classic example that we usually see a lot of is the body temperature. When your body temperature gets too hot, gets higher than 37 degrees, your nerve cells and in your skin and your brain senses that, so it picks up that information. It goes to the temperature sensor, regulatory sensor in the brain that says, hey, you know what, it's too hot, we need to lower it. So what ends up happening is it activates the effect of the sweat glands in the body, and that's going to eventually turn off the body, turn off that heat, so it gets your temperature back down to 37. We see this also in other pathways, like when we talk about breaking down glucose in glycolysis. As you go through the pathway, there's like 10 steps in that pathway. I'm not going to draw them all out. But one of the, when you have too much pyruvate, so pyruvate is too high, what's going to happen is actually going to go back early on and it's going to stop one of the enzymes. So it kind of makes sense. You have too much of the end product. We need to stop what's at the beginning so that we don't continue making too much product. It's like a good, well-oiled manufacturing plant, basically. If you get too many, whatever you're making, let's say you're making chocolate bonbons at the end. If there's too much at the end, well, then we slow down original production so that we don't overflow and make too much overall. Now, like I said, we don't see a lot of this, but there is some positive feedback loops. Here, instead of turning off the initial reaction, what we're going to do is we're actually going to augment it, or we're going to make more of it in the long run. So there's a few examples in humans. Usually you hear the examples in humans for a positive feedback loop. One great one is lactation. If a baby is lactating, it actually tells the mom to continue stimulation of producing milk. And once the baby stops lactating, that actually stops mom's production of milk. But I'm going to use an example also found in plants because I want to remind ourselves that plants are also organisms that sometimes get put in the back burner. And this is an example of ripening of plants. So you probably heard the expression, I'm sure you've heard it, one rotten apple spoils the bunch. The reason why we have that expression is because it is true. If you have an apple that is very, very ripe with a whole bunch of not so ripe apples, you'll see fast the other apples start to ripen and maybe get to that overripe stage. If you ever get a fruit at the grocery store that's not as ripe as you want it to be, you can ripen it faster by just putting it in the bag, in a brown paper bag with a fruit that is already ripened. And again, you're going to see that they'll ripen quite fast. And the reason why is because plants produce a gas called ethylene. And this gas basically is a ripening, it turns on ripening in plants. So let's say you have apples on the tree, starts to ripen. That ripe apple produces ethylene. That gets sends out the neighbors to ripen. The neighbors produce more ethylene, which means more apples ripen. And guess what? We start this over again. It's going to affect the neighbors again. More apples ripen. You're augmenting that stimulus. So that is how a positive feedback loop works. Kind of makes sense why we wouldn't have too much of that in our body, because a lot of times if things are not going right, we want to turn it off. We don't want it to continue. But every once in a while, sometimes we want to continue production of things overall. Now, in terms of signaling, just again, kind of a reminder, in multicellular organisms, plants can talk to each other and to itself in the cells. Some of you guys might have grown cute little pea shoots when you were in elementary school, or some other plants in elementary school. If you had done so, if you had learned to do that, you might have noticed, so you would have something like, here's your plant, little shoot starts growing. What happens is, as the sun produces sunlight, a chemical in plant called auxin starts to produce, lots of auxin. But as the plant is directed differently toward the sunlight, so let's say the sunlight's a little off to the side, you would notice that the distribution of auxin is toward the shadowy part of the plant. And what that does is that causes this side of the plant to get more auxin, which starts cell division. So as this side gets the increased auxin, starts dividing cells more, it becomes elongated here, and you're going to see that bend, that classic bend where our shoots tend to go where the sun is. So if you had that plant at home, if you turned it around, if you moved it the other way, you would notice that all of a sudden the auxin would start to build up on this side, and that caused the elongation on this end, and you would start seeing it bending toward the other side if we move the sun this way. Another way plants talk to each other, believe it or not, is they have defense mechanisms too. So here's, again, a plant that had gotten damaged in one leaf. Well, it might be eaten by some caterpillar or something like that. What's going to happen is this starts to send signals throughout the plant. And those cell signals, those molecules, get picked up by other cells elsewhere. So you might see all of a sudden this signal gets brought up. We have maybe a tyrosine kinase here. They start to activate. The nucleus starts producing some proteins. Here is your some kinases activated. We have ROS pathway activated, which is reactive oxygen species pathways. So all of these proteins are being made in response to this damage, and these are being made in these cells. And what it ends up doing is it starts increasing a type of chemical called gibberellic acid, and that chemical is increasing, and it tastes horrible to bugs. So if there's a caterpillar eating here and it starts to eats up all this leaf, it gets to over here and it's like, ugh, this tastes bad, it will go away. So it's another way that plants can talk to each other, again, using cell signaling, using communication, using molecules to help change what's going on inside the nucleus to make different proteins to help it work overall. Okay, so let's talk a little bit about cell division. In terms of the AP test, you're not going to have to know all the details of mitosis, so I am not covering mitosis. What they really want you to understand are things like the cell cycle. So again, just a quick reminder, our cell cycle is divided really into two phases overall. Those two phases are interphase and M phase. And in case you don't know already, interphase is going to be divided into three more sections called G1, S, and G2. M phase is going to be divided into that prophase, metaphase, anaphase, and telophase. All right, so, and again, what's going on in these phases? So in G1, we have a lot of growth going on. In S phase, which is one of the most important phases, that's DNA synthesis. That is where you duplicate your DNA in preparation for mitosis. Just a quick reminder, you do not duplicate any DNA in M phase. That does not happen at all here. All your DNA duplication occurs in S phase, way before M phase. And then finally we have G2, which is growth and preparation for mitosis. Okay, so just kind of, I think I talked about that already. Just a reminder too, when you're in G1, sometimes you go into G0, that's a non-dividing stage. Sometimes a cell that's in G0 can go back into G1 and divide. Your cells usually do not divide more than 50 times. So 50 times. So after a period of time, it will leave that phase of division and go into a programmed cell death, which is apoptosis. Okay, so again, a lot of this cell cycle is regulated, and it's regulated by two proteins called cyclin and cyclin-dependent kinases. Again, it's basically a particular protein that adds a phosphate group to activate other molecules in the cell to initiate mitosis. They really want to make sure that these, in addition to these, you have these checkpoints that are going to control mechanisms to make sure that you have division occurring properly in those cells. If you don't have this checkpoint, if you don't pass those checkpoints, a couple things can happen. The cell can try to fix itself, which is awesome that it can do that. Or if things are so bad and so damaged, then you're going to go on apoptosis, which is cell suicide and cell death. Basically, it gets rid of itself cleanly, so you don't even realize that that cell ever existed at that point. So things are controlled in the cell by using the cyclins and CDKs. Basically, cyclin levels are going to always change as you go through the cell cycle. So as you go through G1, S to G2 and M, your levels of cyclin will increase as it goes through this, and then it drops off at the end of M phase. CDK stays the same. It doesn't change. It's always there. What ends up happening is as the level of cyclin increases, it's going to bind to CDK. And when it binds to CDK, again, to kind of increase, binds to CDK, etc., it does turn on other genes that eventually are going to help with things like transcription, which you want to do for cell cycle, for the S phase, or also get those proteins ready for mitosis. And of course, that's when everything goes right. Don't forget, when things don't go right in the cell, they can also lead to uncontrolled cell division, and those are your cancers. Basically, this is your cell losing control of the cell cycle. There are two major types of genes that we deal with in cancer. Those are the oncogenes. So these are basically mutated genes that induce cancer. I read this analogy, I thought it was great. Your oncogenes are in your cells, so you probably have some oncogenes, you have proto-oncogenes right now in your cells. Things are going well, things are normal, cell cycle is going well. But when this oncogene gets mutated, it's almost like adding a gas pedal to the cell cycle. We start increasing the cell cycle and we can't stop it. No matter how many brakes we put on it, you can't stop it. You also have some other genes that are working really well right now called tumor suppressor genes. These genes, when they're working normally in your body, as the name implies, they suppress tumors. They're control mechanisms that turn off proteins to make sure you don't have the cell cycle go on when you don't want it to. So normally they prevent cancer cells from arising. But when these guys are mutated, it's like removing those brakes. So normally you have that cell cycle controls checkpoints, something doesn't work, you get rid of the cell, you fix it. Well, now you've lost the brakes. It just keeps on going, keeps on going, keeps on going. So this also can cause cancer in that sense. Of course, if you have these two guys together, that can cause a really aggressive cancer overall. So there's two ways that basically you can genetically induce those cancers. Some environmental factors affect and add mutations to these particular genes. And when you do that, again, that's going to stimulate that cancer growth. That's why environmental factors are important to pay attention if you want to avoid any cancers. If you have already an oncogene in your body, and this you can find out now through some genetic testings, we do have some oncogenes already identified, like the BRCA1 and the BRCA2, then you might want to ensure that you're also not getting any environmental factors, so we can keep this proto-oncogene from becoming an oncogene, or your tumor suppressor gene from getting mutated even further. All right, so that's all we got for cell cycle and cell communication. Good luck.